Welcome to this video in the Patterns in Nature topic. This video will be looking at the first 10 investigation dot point. Perform a first 10 investigation of the movement of materials in xylem or phloem. So up until now, we've been looking at the structures that are involved in transport in plants. So in particular, we looked at xylem and phloem in a previous video and what the structure of those two different vessels are and their functions. So what we need to do is pick one of them and have a look at how materials move through each or just one of those at this stage. So this video is going to be looking specifically at the movement of materials in the xylem vessels. So a way that we can do that is to carry out an investigation called the transpiration in coloured tubes. So the aim of this investigation is to observe the movement of dye through the xylem of celery. So as we know, xylem runs from the very bottom of the plant all the way up to the leaves, and the job of the xylem is simply to carry water. So from that, we can create a hypothesis to be able to look at transpiration. If water moves through a plant via transpiration, if we add dye to that water, the dye will also move through the celery. Obviously, water is colourless, so we're not going to be able to observe how it moves through the celery. But if we add some dye, either red or blue dye, we'll be able to track the movement of that dye through the celery over a period of time and see just how far or how important the xylem and how important the leaves are in the transpiration of water. So what we're going to do is we're going to set up two beakers, both with coloured water. One beaker is going to have a celery stalk that has no leaves on it. And this is going to give us an idea of the importance of leaves in transpiration, which leads us into our next activity. The four celery stalks that have leaves, you want them to be cut all the same length, which we know is one of our control variables. Obviously, we don't want a really short celery stick and a really long one. So you need to determine what the best length is going to be to have all your celery stalks the same length. And then you need to make sure that your celery stalk that has all the leaves removed is also that same length. Then what you're going to do is place all of those stalks into beakers of solution, okay, trying to make sure that the concentration of dye in both solutions is roughly the same. And then you're going to start the timer. And every 5 minutes, you're going to remove one of the celery stalks with the leaves, and using a ruler and a scalpel, you're going to measure 0.5 centimetre increments up the celery stalk until you no longer see the dye. And what you will do is then you'll record the distance that the dye travels in centimetres in your table every 5 minutes until we've gone through all of the celery stalks. When you get to the last celery stalk, you'll also do the same with the celery stalk that has no more leaves, and you'll see if there's an impact or what the impact is of having no leaves on transpiration in xylem. So when you do a cross-section of the celery, we can see these little circular structures around the outside of the celery stalk. So these are actually the xylem tubes, and what you should see is these become stained quite dark with whatever colour solution it is that you are using, okay? So this obviously shows where the xylem is located in the stalks of the celery. We can also cut longitudinally, so down the very outside of the celery, and you will see these darkened tubes running the length of the celery as well. You will also see fairly faint lines in the celery as well, which could quite possibly be, or is possibly, the phloem. So the celery is, the xylem is mostly carrying the water up to the leaves, but then we may also have the movement of some of this coloured water back down the leaves. Okay, once we've gathered all of our results and we have measured how far the coloured dye has travelled in both or all of our celery stalks, we're going to graph that as a line graph. So our independent variable, the thing that we're changing, is the time. We're not changing anything else. We're just seeing how far the dye travels over that time. And obviously our dependent variable is the distance that the coloured water has travelled. Remember, with our title for a graph, we need to have one that indicates both our dependent and our independent variable. So we can just simply turn the two variables into a sentence. So something along the lines of the distance the colour water travelled in celery with leaves over time. So we're obviously not going to be graphing the celery without leaves, because we only have that one, that we're not really looking at a trend, we're just looking at how there is a difference between leaves and no leaves. Okay, so we should get a line graph that really shows that over time, obviously, the coloured water travels further in the celery with leaves. And that brings us to the end of this video, so thank you for watching.